Hello and welcome. This is a science break video for the unit called Bioenergetics. This is for Unit 4 for AQA GCSE Combined Science Biology and Triple Science Biology as well. Timestamps and more information in the description below. Like and subscribe if you can. Other than that, let's begin. We will start off with photosynthesis. Photosynthesis happens in plants. It also happens in algae as well. We have carbon dioxide from the air which enters the leaves by diffusion. We have water from the soil which enters the roots by osmosis. We have sunlight absorbed by chlorophyll which is found in chloroplasts, and then we have glucose that's made in the leaves. We also have a waste product of oxygen which exits the leaves by diffusion. These are the key components in the process of photosynthesis. Carbon dioxide plus water gives us glucose and oxygen. This is a summary word equation. We have energy from sunlight, and we have that sunlight absorbed by chlorophyll which is found in the chloroplasts. This is the formula equation. We can balance this very easily. We just put sixes in front of everything except the glucose. You should be able to recognize and write this formula equation down. We describe photosynthesis as an endothermic reaction. This means energy is transferred from the surroundings to chloroplasts by light. What happens to the glucose that's made? These are the uses of the glucose. It can be used in respiration to release energy for use by the plant. It can be converted to insoluble starch for storage of the glucose. It can be used to make fats or oils for storage of energy as well. It can be used to produce cellulose to strengthen cell walls. And it can be combined with nitrates from the soil to make amino acids. Amino acids make proteins for growth. We'll look at this in more detail in a moment. Limiting factors in photosynthesis. There are conditions that affect the rate of photosynthesis. These are light intensity, carbon dioxide concentration, temperature, and the amount of chlorophyll present in leaves. The effect of limiting factors in photosynthesis. Here are some axes for a graph: light intensity along the bottom, and along the y-axis we have the rate of photosynthesis. This graph looks a little something like this. Let's take a look at this part here, region 1. We say light intensity is the limiting factor for that part of the graph. This is because as light intensity increases, so does the rate of photosynthesis. For part 2, light intensity is no longer limiting. Light intensity increases, but the rate of photosynthesis does not. Carbon dioxide concentration or temperature, or both, are limiting. Let's take a look at another graph with carbon dioxide concentration on the x-axis, rate of photosynthesis on the y-axis. The graph has a very similar shape. For region 1 again, that part of the graph, we say carbon dioxide concentration is the limiting factor. This is because as the concentration of CO2 increases, so does the rate of photosynthesis. For region 2 of the graph, carbon dioxide concentration is no longer limiting. It's no longer a limiting factor. Carbon dioxide concentration increases, but the rate does not. That means light intensity or temperature is limiting the rate of photosynthesis for region 2. If we look at the effect of temperature, that's slightly different. Temperature along the x-axis and rate on the y. We can see the graph has a slightly different shape. For region 1, temperature is the limiting factor. As we raise the temperature, the rate of photosynthesis increases, and that's because the rate of action of enzymes increases for a warmer temperature. As the temperature increases, so does the rate. For region 2, the rate decreases rapidly. The rate of photosynthesis decreases rapidly. This is because enzymes that control photosynthesis denature, which means the active sites of those enzymes permanently lose shape, which means they cannot work anymore. Photosynthesis rapidly stops. The next part is for the higher tier. This is both for combined students and triple science students, higher tier. Knowledge of limiting factors can help growers to keep the best conditions in a greenhouse. Here is the idea of a greenhouse. That greenhouse diagram will disappear shortly, so you have the notes in front of you again. This knowledge helps to maximize photosynthesis, but also to maintain profits. Let's take a look at a graph again. We have light intensity on the x-axis and rate on the y. and here is a curve. Let's say that this is at 20 degrees centigrade with a 0.04% carbon dioxide concentration. We could change the conditions slightly. This one, we have the same temperature, but we have increased the concentration of carbon dioxide. You can see the maximum rate of photosynthesis is a little higher. We can also increase the temperature and increase the carbon dioxide concentration. You can see here that the maximum rate of photosynthesis is at its highest. We have conditions A, B, and C. Important to note that condition C may not be used even though it's got the highest rate of photosynthesis because energy costs may be too high. Also note that light intensity beyond that red line, there's no point increasing light intensity beyond this point because there is no increase in the rate of photosynthesis, so you don't want to have the lights too bright, using more energy and causing more cost. Let's put those notes back up there. Next, we're looking at measuring the rate of photosynthesis. There are various ways we can do this. This is one way. We could take a pondweed plant called Elodea. We can measure the number of bubbles it produces over a certain amount of time, or count the number of bubbles. So we would measure the rate as number of bubbles per minute. For example, 10 bubbles per minute. We can have a slightly different setup. In this case, we are actually collecting the oxygen gas that's given off. That's collected in the upside down measuring cylinder there. And we can measure the volume of oxygen per minute. Not the number of bubbles, but the volume. For example, 3 centimeters cubed per minute. Here is a slightly fancier piece of equipment. Again, we collect the oxygen bubbles that are given off. They collect at the top of the curved bit of the tube there. The syringe can be used to draw the bubble next to the scale, and we can measure the volume. So this measures volume of oxygen per minute, for example, 2 centimeters cubed per minute, or for example, smaller units of 10 millimeters cubed per minute. These are just example rates of photosynthesis. The photosynthesis practical. In this case, I'm going to use the example of counting bubbles per minute, which is something you might have done. We have a ruler scale, we have a lamp, and we set up the apparatus as shown in the diagram. We may add some sodium bicarbonate to the water there. So carbon dioxide is added to the water to make sure it is in excess. In other words, it's more than we need. It is not a limiting factor. We would move the lamp to, for example, 10 centimeters, switch it on and wait for a few minutes. We wait so that the rate of photosynthesis stabilizes, and then we count the number of bubbles produced in a minute. We would change the distance to 20 centimeters and then wait a little while and then count the number of bubbles produced again in 1 minute. We would repeat this for about 5 different distances, no less than 4, certainly 5 or 6 would be good. The graph that we would get would look something like this: number of bubbles per minute on the y-axis to give us an indication of the rate, and distance of the lamp on the x-axis. The graph should curve downwards like this. The number of bubbles per minute indicates the rate of photosynthesis. In other words, it tells you how quickly photosynthesis is happening. The bigger the distance of the lamp, the lower the light intensity, so the number of bubbles per minute decreases. This tells us that we have a lower rate of photosynthesis. Notice the line is not straight and it curves. Control variables for this experiment. These are variables that we would keep the same. We would have the same pondweed, same length of pondweed, same temperature, and this can sometimes be achieved by using a water bath or a heat screen. Same carbon dioxide concentration, or at least carbon dioxide in excess. And we would also repeat the experiment 3 times for each distance to take a mean, and this would give us a more accurate measurement. We can do other experiments as well, which have come up on papers before. We could change the light intensity by changing the brightness of the bulb instead of moving the distance. There's the lamp. We would not move the distance of the lamp. We would just increase the light intensity by, for example, increasing the potential difference for the bulb. We can also change the color of the light that hits the pondweed. We can do this by using filters, color filters. We could place the red one there first. That would shine a red light on the pondweed. We could then change to various different colors, measure the number of bubbles per minute, and record the results for that. Which color light do you think has the lowest rate of photosynthesis? You might be surprised that it's green. Green is actually not absorbed very well by plants at all. It's reflected. That's why plants tend to look green. Now to the fun part, the inverse square law. This is for higher tier students for both combined science and for triple science. The inverse square law looks like this. Light intensity is proportional to 1 over distance squared. That curly little symbol there means proportional. We say this by saying light intensity is proportional to 1 over distance squared. I think it would be better if we thought about it as 1 over the increase in distance squared. We can also say light intensity is inversely proportional to distance squared. I think a better way to think about it is thinking of it as inversely proportional to the increase in distance squared. What does this all mean? Let's take a look at some data. Here is the experiment. We're going to look at an example set of results. Let's start off at 10 centimeters. So the distance of the lamp is 10 centimeters away from the pondweed. We're going to give example data of light intensity of 180 lux. That's a measurement unit for light intensity. And we're going to give an example of the rate of photosynthesis as 60 bubbles per minute. Now, if we increase the distance from 10 centimeters to 20 centimeters, what have we done to the distance? We have doubled the distance. So we have gone from 10 to 20. That's times 2. If we then look at it from our inverse square law, this tells us that light intensity is 1 over 2 squared, which is a quarter. That means the light intensity is also going to be a quarter of what it was. That gives us a light intensity of 45. This tells us the rate of photosynthesis will also reduce, and that will be a quarter of what it was before. So in this case, that's 15 bubbles per minute. Let's take a look at another example. Imagine we change the distance to 30 centimeters. From our original 10 centimeters, the distance is now 3 times more than it was. So light intensity will be 1 over 3 squared, which is 1/9. So if we look at the light intensity row, 180, 1/9 of that is 20. If we look at the rate of photosynthesis, it will also be 1/9 of 60, which is 7. In fact, if you put it into your calculator, it will say 6.7. However, we can't have 0.7 of a bubble. We either have a bubble or we do not have a bubble. So I've rounded that to 7 bubbles per minute. Let's take a look at the rest of the data. You can actually pause here and test for yourself. For example, if we went from 30 to 60, what have I done to the distance? Well, I've doubled the distance. So from above, we can see if I double the distance, I quarter the light intensity. So I've gone from 20 to 5, that is 1/4. And in fact, the rate of photosynthesis has gone from 7 to 2. Remember, we can't have decimals for bubbles. We only have whole numbers for bubbles. So that's the inverse square law. Let's take a look at respiration. Respiration provides energy for processes in all living things. It's exothermic and it happens continuously in living cells. It can be aerobic, where it uses oxygen, or it can be anaerobic, where it happens without oxygen. Let's take a look at aerobic respiration first. Here is the digestive system. Here is the lungs, or here are the lungs. And here are 2 cells very close to a blood capillary. These are our working cells. We have oxygen that's taken into the lungs, glucose is taken into the digestive system in the small intestine, and these are transported by the blood to our cells through the capillaries. Respiration happens, and we have 2 waste products of carbon dioxide and water. We also have the release of some energy. Energy released from this process is used for 1. Chemical reactions that build large molecules, for example, proteins. We have energy released for movement, that's via muscle contraction. We have energy released for keeping us warm. And if you look up here, we said respiration was an exothermic reaction. That's the release of energy, so that actually is used to help keep us warm. Also for active transport of molecules or mineral ions. So let's take a look at aerobic respiration in animal cells, plant cells, and we're going to include yeast cells here as well. The equation for this is glucose plus oxygen gives carbon dioxide and water. This is aerobic respiration in animal cells, plant cells, and yeast cells. This is the formula equation. We have 6's in front of everything except for the glucose. Let's take a look at anaerobic respiration. We're first going to take a look at the example of animal cells, especially muscle cells. Here are two working cells. Glucose is converted to lactic acid and we have the release of energy. We have less energy released than aerobic respiration. In plants and yeast cells, it's slightly different. Still anaerobic respiration, but slightly different. Here's a plant. Here is a single yeast cell. We have glucose that is converted to ethanol and carbon dioxide. In anaerobic respiration, we have much less energy released because the breakdown or the oxidation of glucose is incomplete. We also need a little bit more information about anaerobic respiration in yeast. We call this fermentation. Fermentation is important for making bread. Carbon dioxide released by yeast in bread dough makes it rise. Carbon dioxide, if you look, is in our equation there. Here is some dough that's being used to make some bread. If we add yeast to the dough, it will cause it to rise because of the release of that carbon dioxide, which makes carbon dioxide bubbles in the dough. So, there's the dough. The dough rises because of the yeast, and then after that, the dough is baked to make bread. Fermentation is also important in making alcoholic drinks. Ethanol from fermentation is the alcohol. And if you look in our equation, glucose goes to ethanol and carbon dioxide. The ethanol is the alcohol. The specification wants us to be able to make a comparison of aerobic and anaerobic respiration in plants, animals, and yeast. So we've got aerobic respiration in plants, animals, and yeast. And we've got anaerobic respiration in animals and in plants and yeast. You might want to pause here and give this a go. We use ticks and crosses in the boxes to see if the statement is correct or incorrect. So I'll just go through these. I won't say much. I'll just put in the answers. There you go, a very important summary comparison of those processes. Next, we're going to take a look at response to exercise and metabolism. Response to exercise. During exercise, more energy is needed by the body. To provide more energy, the breathing rate of the lungs increases. This means we have deeper and faster breaths. This is so more oxygen can be taken into the blood for aerobic respiration. The heart rate increases. This means more beats per minute. This means more oxygenated blood and glucose is supplied to working muscle cells. This means more aerobic respiration to release more energy for muscle contraction. This is during exercise. If there is not enough oxygen available, anaerobic respiration takes place. This is when glucose, if you remember, is converted to lactic acid. Glucose is supplied to the cells. There's our lactic acid. This lactic acid causes something called an oxygen debt. We'll describe what that means in a moment. Lactic acid builds up, muscles become fatigued and tired and stop contracting efficiently. This can happen during long periods of heavy exercise. The next part we're going to look at is again for the higher tier. We have our muscle cells working there. During anaerobic respiration, remember, lactic acid can be formed. There's our lactic acid. This is part of the body that includes the liver. There's the liver there. We have blood that will come along and transport the lactic acid to the liver. In the liver, we have lactic acid, which is converted into glucose. This will remove it from the blood. So lactic acid is converted to glucose in the liver. The oxygen debt is the amount of extra oxygen needed after exercise to react with the lactic acid and remove it from cells. It can actually also be converted to carbon dioxide and water and removed in that way. Let's now take a look at metabolism. Here are some examples. Simple sugars in the body can be converted to more complex carbohydrates, and these complex carbohydrates can be converted into more simple carbohydrates, the sugars. We have amino acids. These can be built up into proteins. Proteins can also be broken down into amino acids. We have fatty acids and glycerol. This can be made into lipids, and lipids can also be broken down into fatty acids and glycerol. Metabolism is the sum of all the reactions in a cell or the body. This includes both plants and animals. Let's take a look in slightly more detail. Metabolism examples. This is in plants only. One example is the conversion of glucose to starch. So here are our glucose molecules. These can be converted to starch by being made into a long chain. There's our starch. So glucose into starch. That's one example. Glucose can also be converted into cellulose. So I've got a few more glucose molecules here, and they can be converted into a substance called cellulose. There's our cellulose. Glucose into cellulose. Next, we have conversion of glucose to amino acids using nitrate ions. So here are our glucose molecules. Here are some nitrate ions. These will combine with the glucose and make amino acids. So let's simplify that slightly. Here's our glucose. Here's our nitrate ions. These join the glucose molecules and make amino acids. Nitrate ions combine with glucose to make amino acids. This is in plants and in animals. The formation of lipid molecules from glycerol and 3 fatty acids. Here is a glycerol molecule. Here are 3 fatty acids. These will join onto the glycerol and make lipid molecules. So let's summarize that. Here's our glycerol. Here's our 3 fatty acid molecules. They join together to make our lipid molecule. Next, in animals only, we have the breakdown of excess amino acids to form urea for excretion. Excess amino acids are amino acids that are in the body that are not needed. Here are our excess amino acids. These are broken down into urea, and urea is moved or removed from the body. So we can summarize that like this. Here's our excess amino acids broken down into urea. Urea is removed from the body. We have a final example, the conversion of glucose to a substance called glycogen, which is used for storage in animals. Here's our glucose, and similar to making starch, it's made into chains. So glucose converted into glycogen that can be used to store the glucose in muscles and in the liver. So that's pretty much it. The whole of Unit 4, Bioenergetics for AQA GCSE Combined Science Biology and for Triple Science Biology. Thank you for watching, and I'll see you soon.